Hello friends, this video is consist of about mitochondria protein targeting. We know that mitochondria having double membranous cell organelles. Okay, they consist of proteins are consist of inner membrane, also inside the outer membrane and inner space membrane, interspace membrane, and also consist of matrix. Okay, now we discuss in this video how these proteins are target inside these four regions of mitochondria. Okay, we know that some genome also contains inside the mitochondria. Okay, let us consider it is a nucleus which consists of DNA molecule transcribed to form messenger mRNA, it comes inside the cytoplasm where free ribosomes are present. We know that free ribosomes are present inside the cytoplasm. Okay, these ribosomes attached here and forms synthesize protein molecule. We are having N-terminal end and C-terminal end. It is a post-translational protein targeting. Okay, I have already discussed about post-translational protein targeting in previous lectures, and here some signal sequences attached at N-terminal end. Okay, in N-terminal end having signal sequences. Okay, now there are so many proteins comes inside the mitochondria membrane. We know that mitochondria are double membranous structure. It is a membrane of outer and this one is membrane of inner, and this is the space, inter membrane space, that is IMS. Outer membrane of mitochondria and inner membrane of mitochondria. Here these two lipid bilayers having two different types of transloci. One is one is TOM and second one is TIM. Okay, TOM means translocan of outer membrane and TIM means translocan of inner membrane. Now we draw translocan of outer membrane and translocans of inner membrane. Okay, here Okay, there are so many proteins complex together to form Tom complex. Here Tom complex. Here Tom 20 and here Tom 22 and here Tom 70. Okay, and this one is Tom 70 and Tom 40. This is the main protein which forms channel, and it will create a way to transfer this protein inside this. Okay, and these three are receptor proteins in the outer membrane. Some another proteins are also present. Some another proteins are also present here. Okay, to form Tom complex. Okay, now the TIM. TIM means translocan of inner membrane. TIM complex. There are two types of TIM complex. There are two types of TIM complex here. First one is complex 22 and second one is complex 23. Okay, these two complexes also having other different types of proteins like TIM 54 and TIM 18 and TIM 22. This one TIM 18 and this one is TIM 18 and this one is TIM 54 and this is known as TIM 22. This TIM 22 is for channel forming and this is for TIM 18 for receptor. Okay, like Tom 70 and Tom 20. Okay, and this is for recruitment of protein. Okay, TIM 54. Now the about complex 23. Complex 23. ठीक है. Complex 22. complex 22 target the protein in the inner membrane. Okay, here protein comes. How these actually there are two types of protein produced here. One is cleavable sequence, signal sequence. Another one is having non cleavable signal sequence. Okay, both the protein can target inside the mitochondria, but different regions of mitochondria. This cleavable protein mostly contains STS sequence. What is the meaning of STS sequence? STS sequence. Okay. STS sequence means stop transfer sequence. Stop transfer sequence. When this protein, cleavable protein, interact with TOM22, okay? Cleavable protein always interact with TOM20 receptor, okay? And non-cleavable protein always interact with this. And this one is here, okay? Cleavable protein always interact with TOM20 and non-cleavable protein is always interact with TOM70 receptor protein. Then they convey the protein inside the IMS intermembrane space, otherwise IMS TIM22. Okay. Here there are so many complexes of TIM 9 and 10 or TIM 8 and 13. These are the guide sequences which occurs inside the inner membranous space. Okay, some proteins comes inside the IMS and it convey inside this. Here the protein comes. Yes, some mitochondrial peptidase enzyme cut this peptidase enzyme. This one is peptidase enzyme which cut down this signal sequence of protein. Okay, and this will work in mitochondrial matrix. Finally it will target inside them. Here both the proteins are consist with HSP 70 protein, HSP 70 protein which provide ATP hydrolysis to go inside the outer membrane of translocon, that is means term 40. Okay. And this protein, if this protein comes inside the here, then it will pull together and comes here. Okay. And this also help to comes here. Okay. This pulling of protein here occurs with the help of HSP17, which is cytosolic HSP17, and here also one of chaperone is present, which is also known as HSP17, which also help in pulling of this matrix protein, okay, inside the matrix. And another thing also help gradient. We know that hydrogen ions are more inside the inner membranous space of mitochondria, and here negatively charged are present. We know, all of us, we know this. Okay. It also help in the pulling of this protein inside the matrix. Okay. Now we look at cleavable protein and non-cleavable protein. Cleavable protein mostly go inside the inner membrane with STS. Okay. And non-cleavable protein go inside or outer membrane as well as intermembrane space, as well as inner membrane, and also inside the matrix. Here cleavable protein comes inside the IM, and non-cleavable protein also comes inside the IM. These are not multipass protein. This one is multipass protein. Okay. Here non-cleavable protein is multipass protein which go inside the inner membrane. Okay. This is all about the mitochondria protein targeting. Thank you, friends. Hope you like this video, and we will discuss in next video about chloroplast protein targeting. Okay. Thank you.